Do you want the MRS sign behind you? Okay. And I'm ready whenever you are. All right, you ready?
I'm ready,
Lovely.
yes.
Well, good day, everyone, and welcome to yet another episode of Navigating Nano. Today, I'm your host, Ashley Herring, and I am here today with our guest, Dr. Renee Cottle, out of Clemson University. So I'm so glad that you're with us, Dr. Cottle. Why don't you tell everyone a little bit about yourself?
Sure. So thank you so very much for the invitation. I've never done an interview before, like on YouTube, so yay. Hello, Mom. So as our speaker had introduced me earlier, I'm a professor in the bioengineering department at Clemson University, and I have a lab that is focused on cell-based gene editing therapies for inherited metabolic liver diseases. And so we're actually utilizing CRISPR-Cas technology as part of a therapy to treat diseases such as hereditary tyrosinemia and also familial hypercholesterolemia.
That's pretty awesome. And I was super excited to get to interview you and kind of host this episode because this is very much in line with my research and my advisor, Dr. Eric Josephs. We do a lot of CRISPR-Cas technology and a lot of different avenues, from antivirals and plants all the way to trying to modify and increase the specificity for gene editing as well, hopefully for allele specific editing. So a follow-up question, how did you get into this research? Like, what led you to CRISPR and hepatocytes?
Thank you so much. Yeah, that's a great question. Actually, when I was a graduate student, I was very fortunate to be in a lab that was one of the first to actually even utilize the CRISPR-Cas technology once it was discovered in 2012. So my laboratories was focused on developing a therapy for sickle cell disease, very similar to the therapy that was approved by the FDA in late 2023. So we were actually working on developing non-viral approaches even back then for doing gene editing to treat sickle cell disease. And actually we were trying to do gene correction. Actually, that was the goal, was to correct the disease causing mutation using a variety of different tools. So we looked at TALENs and also zinc finger nucleases. And of course, once CRISPRs came out, we definitely gravitated towards CRISPRs because it was more just having higher efficiency, much easier to design.
Right.
It was very easy to transition over to CRISPRs.
Yeah. And what kind of made you just pick liver cells?
Liver disease. Yeah,
liver disease.
Yeah, that's a good question. So the liver disease part, so as a graduate student, I was focused on sickle cell disease.
Right.
And of course, I wanted to continue working with CRISPR-Cas technology. So when I started my lab in 2016, because I did a postdoc for 1 year, which was, relatively short. When I started my lab, I knew I wanted to continue in the gene editing field and of course, developing therapies. And I wanted to focus on a therapy that would not put me at risk of, competing with my former PhD mentor.
Gotcha.
And then actually one day I was talking to my mom, because my mom is actually, she's a nurse, a registered nurse, and she actually did a lot of research at Emory for the liver transplant division. And so she was focusing on liver diseases and, transplants and even transplanting islet cells. That was one of the studies that they had. And I remember talking to my mom about like what area I wanted to focus on, like what disease area. And she's like, why don't you do something that like treat it? And she was telling me about phenylketonuria. And she was explaining like how there was very limited therapies available, that there's, really no real drugs that were FDA approved for that particular disease. And actually, in fact, the patients are typically treated with a restricted diet and even a drug diet that that would help to lower their amino acid levels in their blood. And so she was like, why don't you develop a therapy that would treat, an inherited metabolic disease like this one? And I'm like, okay. And I decided, so I did the research and I did my own reading and read through the literature. And I was like, sure enough, like she was definitely right. There was very limited therapies available. Like everything that she was saying about the, about that disease plus others that were similar, there was very little therapeutics available. And I knew that would be an opportunity for me to, to develop a new therapy.
Yeah, kind of like a new avenue.
A new avenue of research, yes. Yes. So that's kind of how I got into it.
Awesome. So jumping from sickle cell
disease into inherited metabolic liver diseases.
Also very necessary and needed across the community, of course.
Exactly. And very similar to, for sickle cell disease, there's actually FDA-approved drugs that, like hydroxyurea, that's used to treat patients. But the only curative therapy for sickle cell disease is bone marrow transplantation. And then similar for inherited metabolic liver diseases, the only curative therapy is a liver transplant.
Yeah, which of course are really expensive. And then of course, having to be on immunosuppressants. And
that's
taking a toll. And
so exactly. That's exactly right.
Okay, so now we are over into the liver side of things, and I like that. So I know when you mentioned kind of in your background and how you got interested into CRISPR in general, you kind of started with grad school. Were you always kind of interested in kind of like the bio and things? Like what age did that start? Or
that's a good question.
Is that part of like growing up? And
so kind of like what sparked my interest in science in general?
Yes.
Okay, yes. So I would say that as a child, like I ever since I can remember, I've always been kind of like interested in how things work. And even taking things apart. Even as a child, like I would take things apart just to understand how they would work. And I just naturally gravitated towards science and STEM fields in general. And I remember as a child, like just, watching a lot of Bill Nye, the Science Guy, those type of shows. And even like listening and learning about scientists, like female scientists who've gone to like space, and I just remember always being fascinated with that. And I remember like learning about a scientist, like I remember her name, like Roberta Bondar. She was actually a Canadian scientist, because I don't know if you know this, but I was born in Canada, in Toronto. Yes,
Awesome.
So I was born in Canada, and I just remember learning about some of these Canadian scientists and how some of them started labs when they were young, actually, at home. They would do little science experiments. And so I remember that kind of like led me to want to have my own lab. And I was like, I even asked my parents for like a microscope. So I got a microscope very early on, and then was using it to and I was imagining I was doing experiments. And so,
it's a funny thing. I actually just asked my parents for a microscope for my birthday this past year. So I finally have like my own little lab set up, and the inner nerd in me definitely vibed out with that.
That is awesome.
So
that is awesome. So I had those type of experiences, and of course my parents were very, very supportive of me, wanting to go into a STEM field. And even though my parents are not scientists, and actually my dad is a heavy duty mechanic, and my mom is a registered nurse, so he was not but he was doing things that were technical, highly technical. So
I'm like, because I still think with mechanic you have to think about like how things get put back together. How do you take them apart? They still have kind of like that scientist engineer mindset.
He has the science and engineering mindset. That's so true. Because I remember like when I was a child, I think I was like 10 years old, and I built a circuit with him. And I did that as a project for class. I remember we had to do something science related, and I remember building a circuit, and then I had a whole poster about electricity and circuits, and this is how it works. And so we actually built a circuit. That's really cool. And it was like a pretty significant circuit. Like I had I still remember it had some kind of had a light bulb. It had it even had a siren on it. Like I think my dad had some kind of beeper or something like that.
That sounds really cool.
And he put it on the circuit.
Yes.
And it had a switch too. And so it was actually pretty cool. And I remember all of the students in my class they just loved it. And so things like that my parents did with me, and even to encourage me to continue in this area. And then when I went to undergrad, actually, I attended Dartmouth College, which is an excellent university. I would say most students that were in my class were non-engineering majors or even non-science majors. I would say that the most popular major was probably like econ, like economics, or
Okay.
those social sciences, and so my friends couldn't understand why like I was taking these difficult classes,
Yeah.
But I remember my parents really pushing me and telling me, okay, you need to continue with engineering, even though it's harder than what your friends are doing in school. It's going to open up a wide array of opportunities, and I'm glad I listened.
Yes,
so.
that parental support is always so crucial to kind of like fostering those development and like our hobbies and things like that.
Definitely.
So with that, we know that the parental support is very important. And how was their support and like probably other support influential in getting through the challenges? Because I know there was plenty of challenges. So I guess first, what were some of your challenges of like being a woman in STEM and progressing through that, and then like follow up to that, like what was some of the support to help you get through it?
Thank you so very much. Yeah, sometimes I have to think about it. I'm like, what were the challenge? There's definitely challenges. There's definitely challenges. But I think the early on, that one of the biggest challenges was in undergrad, just being able to having the confidence, especially in engineering, because I was often like one of the only, or very, very few females that were actually taking engineering courses.
Right.
I wouldn't say the only female, but maybe the only female of color that looked like me in the class.
Right.
And so just having to just remind myself, like, you're just as good as the other students. You can do this. And for me, like, it was also an economic thing too, because I didn't come from the prestigious high schools that many of my classmates had come from. So I had to overcome that barrier too.
Yeah.
So I had to just really gain my confidence and really hone in on my skills. And once looking at my grades, and I knew that I was getting my grades were just as good as my peers or even better. It definitely helped to boost my confidence and and I had just reminding myself, okay, you can do this. And of course, having that parental support was very important. Yeah,
and I bet like that confidence build with time helped you get past like that inevitable feeling of like sometimes imposter syndrome setting in and just being oh, do I really do that? But that confidence kind of like helps you feel like I've worked really hard. I do.
That's true.
Belong in this space.
I do belong here. Yes, and then even having the conversation with other females too. So getting support from my peers, because I had friends from a variety of different backgrounds, economic backgrounds, racial backgrounds, and they too also had to struggle with feelings of imposter syndrome or not feeling like they could do it, like they weren't smart enough. And I remember one of my friends in school, like she told me like her father was an engineer, and so he encouraged her to become an engineer. And she told him like, okay, sometimes I feel like I'm just not good enough or I'm not smart enough to do this. And he told her, well, I felt like that too. And so I felt like that was helpful. Like even to hear someone tell me like that her father felt that way.
Yeah.
And I'm like, okay, so everyone feels this way.
Yes, at some point we all feel like, okay, am I doing this right?
Exactly.
Am I gonna make it?
Exactly.
I'm like, do I belong? But yeah, that commonality is just like, okay.
It's across the board. Yeah. Once I realized that, and I think that was a game changer for me. I was like, okay, I'm not the only one who feels this way. Like pretty much everyone feels like this at one point or another in their lives.
So a lot of us, and probably especially a lot of us in the room, all of us in the room really are graduate students. So I want you to think about like, what were some of your favorite moments in grad school? Like, I know between all the work and doing all the research and things like that, there's a need to have like some fun and kind of de-stress.
That is so true. Yes. Yes. It's really important. Yeah. So I would say that I really had a great graduate school experience. It was a great experience. I made some really good friends. And I remember being involved in a lot of like chapters, like we had student-led organizations like B-Bugs, where I was able to like serve on the executive team and get some leadership experience. I also was involved in I'm trying to remember the name, but there's a number of different organizations. There was a Black Graduate Student Association. There was a association for chemical engineers. still forgetting the name. Like,
is it Nobuche?
Nobuche.
Yes.
Nobuche. I almost forget.
Yes, we're
like,
trying
it has a weird.
We're getting a chapter established
Yeah,
here.
Nobuche. We had a Nobuche. Yes.
Okay.
Yes. Yeah, we had Nobuche, and then I was involved in that. I was actually on their leadership. I was the outreach chair for that.
Cool.
So I was the outreach chair for Nobuche, and then I was also on the leadership chair for B-Bugs, and so I was able to do I involved with a lot of different outreach activities where graduate students went into inner city schools and did science demonstrations, and it was a lot of fun. I went with friends of mine from the program and even from other majors that came along with us, and so that things like doing activities like that were always a lot of fun.
Because I know that kind of helps put the whole grad school experience kind of in this perspective a little bit. It's just like, okay, I know we want to do this, of course, to get a degree and then get a job, but also what does all of that mean? And I think by doing sometimes some outreach and joining different clubs, we have to remember that, okay, we're also kind of doing this to inspire younger people.
That's so true.
Younger generation that, hey, you can do science.
That's true.
As well, you can find your place in this field.
I think that's really important.
Yeah,
it's really important because like when I was when I think about it, when I was in the K through 12 level, I don't recall ever meeting a graduate student that, like, to be honest, I don't remember meeting a graduate student at all.
You know what? I don't know if I do either.
I remember meeting, professionals like engineers, but not like graduate students.
Yeah.
So I think that it would have been helpful, to have those, to be able to have those type of interactions to let students know early on, okay, this is an option.
All right, cool. So we do have, of course, some grad students in our audience as well, and we want to open up the floor to them to have them ask you any questions that they may have on their minds. So if anyone has questions for Dr. Cottle, this would be a great time to ask them.
Thank you. I enjoyed your presentation and also this interview. And I'm wondering if you have some kind of workshop in your lab for grad student that we can join, for example, for 1 week or 2 weeks to only observe the process of what you're doing for CRISPR-Cas.
That's an interesting question. That is a great question. Unfortunately, I don't have any workshops at this time, but that is definitely something that I want to develop for sure in the future.
That would be great.
For workshops. Yeah, that's a great idea. Thank you so much.
You're welcome. Thank you.
Hi, Dr. Cottle. I really enjoyed your presentation and I've enjoyed the discussion so far. And
thank you.
This is, the Black History Month. And looking at you, looking at me, I can relate. I just want to know, being a Black woman in STEM and doing this exceptional work that you're doing, how do you include the DIE in the work that you do? Because I am assuming as a professor, there is an aspect of outreach that you get to do.
Oh, yeah, definitely.
So being this iconic month where we get to celebrate amazing Black scientists, how do you get to present that to either in STEM or, with your community?
Thank you so much. Yeah, I really, that's an excellent question. So of course, Black History Month is very, very important. I definitely acknowledge, the importance of it and outreach. So for me, I actually, do my best to do outreach activities throughout the year, especially during the summer months, because that's when I have more free time. And so there's actually, we have Pure and Wise initiative at our school. So it's actually an organization that does a lot of outreach for students of color or even first generation students. And I do like a summer class. It's like a 3-day course for middle school aged science females, actually for females in STEM. And so I actually teach a class on genome editing, and I like, for me, that's like the best way for me to go back, to reach back and interact with students of color. And there's actually a large percentage of the students are involved in that outreach activity are students from minority, underrepresented backgrounds. At this stage in my career, I don't have any REU programs that I'm specifically leading or developing for underrepresented minorities, but definitely in the future as I get more established, that is my goal, is to be able to do more REU programs to inspire others that are that are underrepresented in the field.
Yes. I had another question. Looking at the work that you do, CRISPR, it's expensive. And you working, doing in vivo studies, I can only imagine how much that goes into the work that you do. In terms of, we are graduate students. Some of us would want to be professors at some point and do our own research. So in terms of acquiring funds for the work that you do, how is that process like? How rigorous is it to really secure enough money for all the objectives that you actually laid out today?
That's a very good question, and to be honest, these are questions that I wish I had known when I was at your stage as a graduate student. But it is very competitive. I would say that it is competitive getting funding, but don't be discouraged, because if I can get funding, I believe you can too. Because it really takes a lot of persistence. So when I first started my lab, I did get startup funds, and I would say that most professors get a startup package. Some packages are stronger than others, to be honest. Not all schools have access to unlimited amount of money. It could be difficult. It's a difficult time for institutions. But then taking that startup funds and then using it to get started, like purchasing equipment and then performing preliminary experiments is the next step. And then applying for grants. Like there's, for me, I applied for like career development awards. So including the Pinnacle Research Award that I received for the AASLD Foundation. So I applied to foundations like that. And I was successful. So I would say I was successful with that. I also applied for grants that were competitive in my state. So the South Carolina INBRE. So only investigators in South Carolina were applying for that. And again, that's a career development award. And then even internally, there was awards that I can apply for within my department that was extremely helpful, including the South Carolina Biocraft. It's a COBRE that provided funds for my lab. And then as I was applying for those funds, I was conducting research because I was getting grants. And once I had enough preliminary data, I was able to submit an R01. So it did take me, for me, it took a little bit long, I would say, longer for me to get grant funding, not excessively long, but it did take a little bit of time to get enough preliminary data to be competitive for R01 funding.
Okay. My last question. In terms of mentorship, you did mention your parents, and they seem to have been very great mentors for you.
Yes.
How has been that mentorship journey for you when you were a student, and what are you doing right now as a mentor?
That's a good question.
For the graduate students.
Yeah, that's a very good question. So as a graduate student, my mentorship looked like, of course, I had my PhD mentor, and I also had collaborators, my PhD mentor's collaborators that I remember there was one in particular that I worked more closely with because I would often have to use their lab space to do some of my experiments. And really the mentorship consisted of me just, communicating with my mentor and sharing the research progress with them. And I would say that it was more like professional, it wasn't a lot of career development happening, but my mentor did encourage me to stay in academia. I remember the conversations that we had about careers. I remember being encouraged by my PhD advisor to stay in academia. Because he told me that he felt very strongly that I had the capacity and I had the skills to be successful.
Right.
So it definitely helped. That was very, very encouraging.
I wish I had somebody to like tell me if I should stay in academia or go to the industry. But you do. Like,
yes, I did have someone do that, and actually, to be honest, I'm very grateful. I'm glad that you brought that up because it did remind me that I did have people even outside of my parents encouraging me to stay in academia, including my PhD mentor. And then it definitely having someone like that, telling you like, okay, you can do it too. It means a lot. Yeah. It really does.
Okay.
So this is a more technical question. I didn't get around to asking at lunch, but so I know there's a clinical trial going on right now that uses like a device to implant pancreatic stem cells to try to treat type 1 diabetes, and the device like helps the cells from being helps shield it from attack by the immune system. So I was wondering if there's like a possibility of doing something like that with the liver.
Oh, okay. So from being attacked, that would be interesting. I would definitely. I think that there's definitely a possibility. I would have to do a little bit more research into how that device could be adapted for the liver. I think that would be a little bit challenging to insert a device like that. But I think that would be a really interesting project to look into, for sure.
All right. Thank you.
You're welcome.
I guess I have a question. Going back to your bit again, I wish I could ask a more technical question, but a lot of biology is a lot of acronyms that are confusing to me.
Okay.
I'm more than you side, but I wish I understood. It's always cool seeing you guys talk about CRISPR, BNA, and all this other stuff. And it's like you guys understand each other. But it's like,
that's funny. Yeah,
it's cool.
Thank you.
But I guess, and that just generated a second question. But I think I'll answer the second question I just generated first. So, I guess some of this language barrier I just described, like how is it like? How do you kind of navigate that? And especially if you are kind of expanding this project, you're gonna have to interact with
That is so true. Yeah.
How would you probably just know just enough, or just kind of trust people. Okay, you know what you're talking about. Just kind of do it for the project.
I think that what you're trying to. That's a really interesting question because I think what you're getting to is like how do you navigate the barrier, like the language barrier between like scientists and the clinicians? Is that kind of
or just anyone who's a few degrees from who's not part of the same discipline you are and don't speak
That's
the same.
true. Yeah.
Just anyone, so.
That's true. So for me, like what I try to do is I try to study the audience. Like if I go to any, into any forum, or if I'm meeting with people, I try to study, okay, what is their background? What are their limitations? And then just trying to really break down my project into very elementary terms so that they can understand it, especially if I'm talking to someone who's in business or not even in science at all. And so I try my best to to be able to articulate my science in a very basic way. And in that way, everyone can understand what I'm doing.
Right. So when you came today, I guess when you made a presentation, what did you understand your audience would consist of?
That's good. That's a good question. So I felt like, so I knew that this is a nanomedicine center, nanoscience. So I definitely expecting students to have some basic level of biology background, chemistry background. Is that true?
More or less.
More or less. Most, I would say 50%, more than 60% of y'all have taken a biology course. So if you at least 60% have taken a biology course, I felt like they would at least have some level of understanding about gene therapy and would have heard about CRISPRs, and so I try to design the presentation in a way that that they can kind of get an idea of what, in a sense, and appreciate what we're trying to do in terms of a therapy.
Okay.
And so was there anything in particular that you didn't get? Or I guess I know that you were trying to understand the metabolic pathways. Or
Yeah, I want to expose myself, but I always I think I was explaining to Faith earlier. I'm like a traditional engineer, right? So it's like, okay, what's the application? But you were talking about kind of the application.
So the application would be a therapy.
So when you started talking about the application, I was like, okay.
Application, yeah. So the application. So if you think about therapy, we're trying to use CRISPRs as part of a therapy, and what we're trying to do is treat. We're trying to treat the disease at the genetic level, right? That's essentially what we're trying to do. So these are genetic diseases that are caused by mutations in genes that are essential for the liver functions. So one of the liver functions is to be able to regulate different proteins that are in the bloodstream, or even regulating the metabolism of metabolism. So that it has there's a lot of metabolic functions that the liver has. So if you have, for example, an amino acid that you've taken up from food that you've ingested, the liver is going to break down those amino acids, so that way your body can excrete it and it doesn't accumulate in the blood. So what we're trying to do is we're trying to treat a disease that causes toxicity in the liver by reprogramming, pretty much reprogramming cells outside of the patient's body and then putting them back in, and then having them engraft and repopulate the liver.
Okay,
yeah.
That's cool.
So the idea is to be able to take the patient's cells. So if you remember that chart that I had shown, if you go back to at the very beginning, I had shown this schematic, almost like a circle.
Yeah.
So it starts off with the liver, and the idea is to be able. So if a patient that has a disease, right, we want to treat the patient's disease, but using cells from the patient, and we need to take cells from the liver specifically. So what we want to do is to be able to take resected liver. So we would take a segment of the liver, a small segment of the liver, maybe 12% of the liver, cut that out, extract cells from that segment, and then perform engineering on the cells using CRISPRs to engineer the cells, to treat the cells, and then we're going to be transplanting those cells back into the patient.
Right. So
that's pretty much the life cycle.
That was the.
You understand that, yeah?
Yes, I understand that.
So the part of the life cycle that I was going into was, how do we do the engineering? That engineering part, you think about it, the engineering that goes into, into reprogramming the cells to treat them. That's the black box. And what I was trying to explain was what's going on inside the black box to make that work. So there's a lot of work that's involved, including optimizing the delivery of the CRISPRs. Well, first of all, designing the CRISPRs, designing them, and then optimizing the delivery, validating that it actually is doing what you want it to do. That's what I was presenting today.
Right.
I was presenting data that shows the efficiency of the gene editing. We also showed that we can, we did see some off-target effects, at least in one gene. And so, but it's fairly specific. And then from there, we did the transplantation. So that's putting it back into the patient. So we did this in a mouse model, and we showed that the mouse, I think I showed a schematic of how we injected the mice with the cells. And then we monitored the cells. We monitored these mice, these transplanted recipients, and then we evaluated whether we corrected the disease. And that was the bars that I was showing.
Okay.
For the different biochemical markers. We showed, you want those markers to be low, not elevated. So it's elevated in the disease mouse, but the biochemical markers are lower, the liver biochemical markers are lower in the transplanted recipient mice.
Yeah, so this, I guess, method needs to be used in conjunction with the popular pharmaceutical for liver disease?
Yeah, so the pharmaceuticals for the liver, so currently liver transplantation is the only curative therapy that's available readily for, at least I would say for patients that have liver diseases, but there's severe shortages of liver of donors donor organs, and so it's actually not used as much as it should be because there's such a severe shortage. And I think I talked about a little, like in the presentation, I was talking about gene therapy. So gene therapy, there's currently a lot of development on novel alternative approaches to liver transplants, which would involve using viral delivery systems to deliver like the therapeutic transgene that's deficient in the patient. So for example, in hemophilia, you have the factor 8 and factor 9 that's deficient, and these are factors that are needed for clotting. And so there's actually been a new therapy that was approved for introducing that factor that's needed by the patient for the blood to clot. And so, what we're trying to do is similar to that approach, but we're doing in ex vivo gene editing and then combining that with hepatocyte transplantation. So that way we're not exposing the patient to, viral particles that can have potentially, some side effects, some severe side effects, including the immune responses, even tumorigenesis, those types of risks.
Okay.
A couple more questions.
Yeah. I'm curious if you ever investigated the allelic heterogeneity in the liver tissue?
Oh, what was that? I'm sorry.
Heterogeneity.
Heterogeneity.
Yeah.
Oh, like mosaicism or
For example, the disease resulted from various mutations.
Oh, mutations.
Different
Yeah,
cells
interesting.
of the
Yeah,
tissue.
that's a good question. So for hereditary tyrosinemia type 1, the mouse model that we used, it was a knockout mouse model. Mm-hmm. So it doesn't have any expression of that deficient that enzyme that's involved in the tyrosine catabolism pathway. Mm-hmm. What we're trying to do is reprogram the cells so that way, regardless of what kind of mutation they have, you would be able to correct the disease or rescue the disease in a mouse, or even in a human patient potentially. So the heterogeneity of the mutations would not impact the therapy that we're currently developing for hereditary tyrosinemia type 1. For familial hypercholesterolemia, it's also the same thing. What we're trying to do is develop. So I didn't talk about the new therapy work that we're doing. We're focusing on familial hypercholesterolemia in my lab right now, and this is as a more common inherited metabolic liver disease, and it actually can cause atherosclerotic cardiovascular disease, and it's characterized by high levels of cholesterol. And so what we're trying to do is we're trying to lower the cholesterol levels by pretty much by targeting a gene that when it's knocked down, you would get lower cholesterol levels in a patient regardless of what kind of mutations they have in the LDL receptor gene.
That's interesting. Thank you.
Mm-hmm.
I'm sorry, Ashley. I have so many questions. I enjoyed your discussion today, your presentation today. One thing that fascinated me is the clonal expansion of the cells that you were doing. And just seeing you injecting 1,000 cells, which would typically expand into form an entire organ, that was good. That was fascinating.
Oh, that was actually, I was showing it was a schematic from a paper. Mm-hmm. So that paper showed that the minimum number of cells that you need to transplant, healthy cells, starting with healthy cells, would be 1,000 in that particular mouse model for the hereditary tyrosinemia, specifically for that disease. But that's not, I wouldn't say that's relevant to all inherited metabolic liver diseases, specifically for that one in the mouse model. Yeah, in the mouse model.
Yeah, so I got curious because you're using mouse models. Now, technology has evolved into like tissue models where you can have a tissue model in a in a microfluidic system or like. So I was just curious if you foresee a future where you can have your system, not using the mouse model, but have a model where you have the kidneys, you grow the kidneys and the...
Interesting. Okay.
Yeah, and then monitor, because I think one of the questions that came up was how are you monitoring if the cells are not moving
going
to
to
other
are not
organs,
localizing
right?
to other organs. That's a good
Yeah.
question. So one way to monitor would be to do like cell tracking. Where you can put, for example, like some kind of fluorescent label, like, or even like a fluorescent label, and of course it would have to be sensitive enough where you can detect it and do like live imaging, or even dissecting out the tissues after transplantation and evaluating whether you have those the cells that you transplanted localized to those organs.
Okay.
That would be I think that would be a great study. I know that actually there was a study that I had read recently, and it was it was about the AAV delivery system. And this system, they actually use a fluorescent marker. It was like a TD tomato, something like that, a fluorescent marker, to be able to evaluate where the cells were localizing, or where the cells that were uptaking this lipid nanoparticle. And they showed that there was fluorescence in different organs, including the heart, the liver, the kidney. Yeah, they even looked at the brain, but there was very low amounts in the brain, but there was actually localization in different places.
Yeah.
And that was from a lipid nanoparticle delivery system.
So that may be happening in your cells. Lastly, because I can see we are out of time. CRISPR editing systems, are you using them for therapy? In terms of translation, now, long term is to see it being used in real patients. And this can be in two different questions. One could be the ethics of using CRISPR. How do you think at your standpoint right now, the ethics of using CRISPR as a treatment in humans? Because we have seen it being translated for certain diseases, but what is your stand in seeing your system being used in a real human being? Number two, how long do you foresee your CRISPR editing system being used as real therapy in metabolic diseases?
Oh yeah, that's a good question. First I will address the question about the ethics of what we're doing. So where I stand is actually I feel like what we're doing in the lab consists of doing somatic cell gene editing, and that involves doing gene editing in adult cells. So these are cells that are not going to be used to give rise to, like a baby, a pregnancy. So germline gene editing, on the other hand, would involve doing gene editing in the germ cells, which would then change the genome of future generations. And that's where I draw the line. Personally, I don't agree with germline gene editing. I still think that it should be banned. And it has been banned I would say internationally, although there has been the CRISPR gene editing that was done to give rise to a pregnancy in China a few years ago. Yeah. I'm not even sure how old the patients are at this point, but that was definitely illegal. And I hope that scientists never try to replicate something like that again. But gene editing could also provide a lot of benefits for biological experiments that can be done even in embryos, just to understand, embryogenesis, or even to understand human development. So I think that keeping it for specific, like for very basic genes, experiments, or basic experiments for understanding human development, it would be beneficial, but not for giving rise to pregnancies or,
So I want to thank our grad students, Faith, Gerald, Mabube, Hillary, for all of their wonderful questions for Dr. Cottle.
Thank you so much.
And
those are great questions.
To wrap out this discussion, because we're right near the end, what we like to do is that a previous speaker will pose a question for our next speaker.
Sure.
And so what that speaker's question for you, as we wrap this up, what skill set or skill sets should a graduate student learn or get during their term, during their studies?
That's a very great question. So the skill sets. So of course you want to have, your technical skills, like how to do, assays in the lab, or even, techniques is very, very, very important. But then also the soft skills are also equally as important, like how to deal, how to communicate, how to present your research.
Yeah.
That's very, very important.
Yeah.
How to be comfortable, when you're in a setting, like for example, a conference, like how to go to a conference and get, derived benefit from your attendance at the conference.
Yeah.
How to apply for grants. That's a soft skill. And it also is a technical skill, because you have to have, some technical knowledge to be able to develop a grant idea.
Right.
But then just how to write a grant
Yeah.
is something that's really important. And how to write papers
Yes.
and abstracts and all
Yeah.
those things. So soft skills like writing, communication, oral communication, written communication, and even leadership skills, like how to manage a project.
Right.
Like there's some project management skills that's really important as well. And it starts off by just managing your own project and being able to communicate and interact with different key personnel that are on the project, like not just only your advisor, but you may have to reach out to your collaborators. Because when I was a graduate student, I remember emailing my mentors, collaborators, and even sitting down with them and explaining what the progress was on our project, even without my mentor, my PhD advisor, being present. And so I think those types of skills are really important, and that it would help you or set you up for being successful even outside of graduate school.
And even though this is Navigating Nano, I think that last piece is something that no matter the background or study of anybody listening to our podcast can carry over into their field. We're all going to need to know how to write well, communicate well, and of course the technical skills we're going to need to do our jobs once we're done with grad school.
And even mentoring other people.
Yes, mentorship.
Mentorship. Like I know a lot of graduate students, they have the opportunity to mentor undergrads. And if you have an opportunity to do that, I highly encourage you to take advantage of that experience.
Awesome. Well, thank you so much. I'm so glad that you were able to sit and talk with us.
Thank you so very much. It was fun.
So thank you for joining and listening to this episode of Navigating Nano. Next week, or our next episode, we are going to have Dr. Daryl Boyd from the U.S. Naval Research Lab, who is going to be doing some polymers. So be sure you tune in for that next episode too.
Thank you.